in the next several segments, what we are going to do is go through several different bioanalytical techniques that are particularly useful in the field of chemical biology, techniques that are particularly useful in using chemistry to understand biological systems. So what I'm going to do in this video is just go through an outline of those techniques that we're going to be looking at in a little bit more detail in the upcoming videos and give you kind of a broad framework of what types of structural information each of these techniques will provide, so that ultimately what we are doing here is assembling a full toolkit of methods that will ultimately allow us to determine the chemical structures of molecules that are relevant in biological systems, getting at that crux of chemical biology, of using chemical tools and chemistry to understand how biology works. So our list of bioanalytical methods, in no particular order here, include UVVIS, spectroscopy, where what we are looking at in UVVIS is the fact that select functional groups will absorb radiation within the UV visible range of the electromagnetic spectrum. And those select functional groups are often ones that have conjugation, meaning alternating patterns of single bonds and double bonds, or pi bonds, are very noteworthy for their absorption within the UV visible region of the electromagnetic spectrum, which ranges from about 190 to about 800 nanometers. And as a result, the UV-Vis can provide us with evidence of certain functional groups based on the wavelength at which UV visible light is absorbed. Now keep in mind, while it can provide evidence of certain functional groups, it's not going to give us information about the overall structure of a compound that was completely and totally unknown. Instead, we need to merge this information with other bioanalytical methods in order to develop a complete understanding of the chemical structure of the unknown molecule, such as an unknown drug molecule or unknown biomacromolecule, such as a novel protein or something like that. So we can combine UV-Vis with other techniques, such as IR, which is looking at the absorption of electromagnetic radiation in the infrared region of the spectrum. Specifically, what it is looking at is the wavelength region where bonds will bend, stretch, and vibrate. And that bending, stretching, and vibration is in association with particular functional groups that are present. So what we are looking here is at the wavelengths at which specific functional groups are going to be observed with bond bending, stretching, or vibrations. And those bending, stretching, and vibrations are going to appear at certain regions of the IR spectrum. And empirically, we can know from historical information that specific functional groups will appear at specific regions of the IR spectrum. And so as a result, there's a variety of tables that exist for matching specific functional groups with specific absorption ranges in this IR region of the spectrum. And so we can very readily pick out such functional groups as carbonyl groups, alcohol groups, and others by looking at the IR spectrum for a particular molecule. So again, like UV-Vis, this isn't going to tell us the complete structure of a molecule, but it can start to allow us to deduce different functional groups that are present in the molecule as one of the puzzle pieces for assembling the complete structure, because ultimately determining the complete structure of a molecule is reliant upon generally a variety of different types of information or a variety of different puzzle pieces, making determining the structure much like puzzle solving. So as we're wrapping our mind around bioanalytical chemistry and its applications, you can really think of this as molecular puzzle solving. So another piece of the molecular puzzle that we can use to determine the structures of organic molecules is we can use X-ray crystallography or X-ray diffraction. In X-ray crystallography, what we do is create a crystalline structure of our molecule of interest, and then we subject it to X-rays and measure the resulting diffraction pattern, which gives us information about the electron density of the molecule, and from that information about electron density, we can infer the spatial location of atoms, since atoms are going to be in regions with electron density, since the electrons are there within the atom. So X-ray diffraction and crystallography is going to use X-ray diffraction patterns to determine regions of electron density within a molecule. And those regions of electron density allow us to deduce the atomic structure, and in more complex molecules such as proteins, in many cases the complete molecular structure can be determined. So X-ray crystallography and X-ray diffraction is a very powerful tool, because that's the first tool of this lineup that we've mentioned so far that in many cases can enable an experimenter to determine the complete structure of an organic molecule, rather than just determining that there are select functional groups in the molecule. And so that makes it really powerful. But as with most powerful techniques, there are some limitations and caveats. The biggest limitation that comes into play frequently for X-ray crystallography is that it requires that you be working with a compound that you are able to generate crystals of. And so it is not applicable for liquids, for example, because liquids are not crystals. It is applicable for proteins and nucleic acids if you can crystallize them. Some proteins are readily crystallized, others not at all. And so that has to be kept in mind as a limitation of this particular method. But if you are able to crystallize a molecule, meaning a small organic molecule such as penicillin or cholesterol or some other organic molecule, you can oftentimes determine the complete molecular structure of this, including the stereochemistry, including the configuration at all of the stereocenters in the molecule. So that makes it a very powerful technique, but only in cases where the compound will crystallize. So going on down the list in our arsenal of bioanalytical methods, we next come to mass spectrometry, which we abbreviate as MS for short, for mass spectrometry. Mass spectrometry on our summary list here, or overview list, is a technique in which we ionize molecules, and then for those ions, we detect the mass to charge ratio. And from the mass to charge ratio of that ion, we can backtrack to deduce what the anticipated molecular weight of that compound was. From the molecular weight, we can determine the molecular formula candidates. So mass spectrometry, what we do in this is we determine the mass to charge ratio. We abbreviate mass to charge as M over Z, where M is the mass of the ion. Z is the charge of that ion, oftentimes +1 or -1. So we determine the mass of the charge of the ion to deduce such things as the molecular weight of the compound and come up with candidates for the molecular formula, that is the number and type of atoms that are in that molecule. So this can be a very valuable technique for determining the molecular formula. Much like UVVIS and IR though, in most cases you can't determine with certainty the complete chemical structure of the molecule based on its mass spectrum. Instead, you get information about the molecular formula and maybe some preliminary information about the structure. If, for example, you use the mass spectrometry experiment to break the molecule into fragments, you can analyze the mass to charge ratio of some of those ionized fragments to start to piece together the structure. But usually with a mass spectrum, you can't determine the complete structure of a molecule that was initially totally unknown in structure. So usually the full structure will remain a bit ambiguous unless you have some standards to compare your mass spectrum to, or your compound of interest is very closely related to a previously known compound. Mass spectrometry has very much revolutionized understanding of chemical biology in the last decade or two. And so as a result, we will be spending quite a bit of time in this unit later on the ins and outs of mass spectrometry, so stay tuned for that. Another technique that is extremely important that we are going to be spending some time on later on in this unit is NMR spectroscopy, which stands for nuclear magnetic resonance. In a nuclear magnetic resonance spectroscopy, what we are going to be doing is evaluating connections between atoms to assemble the full structure. So specifically, we're going to be detecting nuclei of specific nuclei that are able to participate in this phenomenon of magnetic resonance, which is 1H, meaning the most abundant isotope of hydrogen, carbon-13, not the most abundant isotope of carbon, but still prevalent enough for detection in many cases, nitrogen-15, phosphorus-31, and there are other atoms that are suitable for this nuclear magnetic resonance as well. These are the ones that are most prevalent when thinking about biological applications of this. So those are the ones that we're going to focus on here. But we're able to detect those atoms and use the resulting spectra to determine the connections between atoms. And by determining the connections between atoms, ultimately, we can determine the complete chemical structure. Particularly if we use this NMR data in conjunction with the other types of data listed as above, such as the mass spectrometry information. So for example, if we can use mass spectrometry to come up with candidates for molecular formula, then we can take that molecular formula and use the NMR data to determine the actual connections between those atoms that compose the molecular formula. Or we can use the IR data and or UVVIS data to piece together some select functional groups in the molecule, such as determining there's a carbonyl group in the molecule, and then use NMR both to confirm the presence of that functional group and also determine exactly where in the complete chemical structure is that particular functional group. So NMR is a very valuable technique. We are going to be spending most of this unit on NMR, looking at both the traditional one-dimensional NMR experiments that we covered in the organic chemistry course, that being proton NMR spectroscopy, and also carbon NMR spectroscopy. But we're also going to get into the more powerful two-dimensional NMR experiments that allow us to directly relate which carbon and hydrogen atoms are directly bonded to one another, and also which hydrogen atoms are in the same spatial proximity as other hydrogen atoms, and things like that to come up with a complete understanding of the full structure of a molecule. So we will get into the details of interpreting NMR data, and by the end of this unit, you should have a much better understanding of applying both 2D NMR and 1D NMR together to come up with the chemical structures of relatively complex organic molecules, and also synergizing that information from NMR with these other tools and techniques that we have looked at. So that's where we're headed in this unit. And in the next several videos, what we're going to do is go through each of these techniques one at a time to look at them each in a bit more detail at the level of detail that you need to be familiar with in order to determine the complete chemical structures of unknown molecules that are relevant toward chemical biology.